These notes cover the tail end of Chapter 7, Section 2 of your textbook, which covers the composition of the cell membrane and its function. Starting with function, the cell membrane's job is to regulate what enters the cell and provides protection and support. Now, the fact that it lets some things in and some things out is called selectively permeable. Which essentially means that if you've got the membrane, some molecules are allowed to enter, while other molecules are not. So that's what selectively permeable means. The cell membrane is also a flexible boundary between the cell and its environment. And the key there is flexible because it's the molecular composition of the cell membrane is mostly lipids. And lipids are flexible. Now, that said, some cells have a cell wall which helps to provide rigidity and actually kind of counteracts that flexibility in a way, but animal cells don't have cell walls, and so their flexible boundaries can be felt because if you actually touch your skin and you kind of provide a little bit of pressure, you can feel that it moves, right? You're squishy. The cell membrane structure, as mentioned before, is mostly made up of fat, and that's the same thing as lipids. And remember that the two components of a fat are glycerol and fatty acids. Now, the very specific type of fat in a cell membrane is called a phospholipid. It's not a triglyceride, which is a glycerol and 3 fatty acids. A phospholipid has 2 fatty acids. And instead of the 3rd fatty acid, there is a phosphate group attached in the 3rd slot on glycerol. A nice depiction of that is here. So I'm going to use my highlighter, and I'm going to highlight the glycerol, and I'm going to label it. And I'm going to take another color, and I'm going to highlight the fatty acids. So you've got 1 fatty acid, 2 fatty acids, and I'm going to label them. But what was a 3rd fatty acid is now a phosphate group. And phosphate has a chemical composition of P, which is a phosphorus, and 4 oxygen. So whenever you see PO4, you know that's phosphate. 1 phosphorus and 4 oxygen. Now, you might remember that fats are nonpolar. However, the addition of the phosphate group causes the glycerol and the phosphate region to become polar. So I'm going to label that. It says polar head. Now, what the heck is the polar head? The polar head is the glycerol and the phosphate group. So I'm going to circle that, and I'm going to draw an arrow, and I'm going to draw an arrow over to where it's called the polar head. The polar region of this fat is now attracted to water. Water loving. The 2 fatty acids are still nonpolar, and so they are water hating, and another term for that is hydrophobic. The polar head is hydrophilic. So you now have two regions, a polar region and a nonpolar region. What ends up happening is that because you are mostly made up of water, the tails will orient themselves away from where the water is. So namely, that's going to be outside the cell and inside the cell. So the polar heads will orient themselves tails in and polar heads away. So here you can kind of see the tails. I'm drawing two at a time. And then the phosphate group and glycerol are up top. And the same thing down below. So cell membranes actually have two layers of phospholipids. One is oriented right side up, and the other is oriented upside down. Now this picture here is supposed to represent a cell, as essentially represented as a ball. And we're not even looking at the contents of the cell, but if you look really closely, you can see the phospholipid bilayer, okay? So phospholipid is each one of these molecules, and I'm going to circle just one phospholipid. And then the two layers we call a bilayer. So cells have a phospholipid bilayer. Now, it's not just fat that makes up the cell membrane. There are other parts. Another component of the cell membrane is cholesterol. You might have heard cholesterol being bad for you, and it can be bad in your food in excess. However, you do need cholesterol embedded in the cell membrane because it helps to maintain the proper fluidity and permeability of the cell membrane. Now, cholesterol is a waxy fat-like substance. You don't have to know the exact chemical composition of it for this class. But essentially, the model of cholesterol is represented here, and they've labeled that. And you can see how cholesterol is embedded in between the fatty acid tails. Just take a note of actually where it sits. It's essential for the proper, as mentioned before, permeability and fluidity of the cell membrane. So we have fat, we have cholesterol, and finally, we have protein. Cell surface proteins provide many of the jobs that the membrane will need in order to allow the cell to function properly. So what I mean by that is you've got proteins that are going to allow things to enter and exit the cell. You've got proteins that are going to trigger reactions by the cell. You've got other proteins that are going to identify the cell. So in addition to lipids, cell membranes contain protein molecules that are embedded in the lipid bilayer. The three functions that I just mentioned are to move materials in and out of the cell, and they are called channels. I'll come back to that in a second. You have others that respond to chemical signals. They're called receptors. And finally, you have some that help to identify the cell. And they're called markers, or glycoproteins. I'm going to cover them in the order that I've just listed. So let's start with channels. Channel proteins are donut-shaped, meaning they look literally like a donut. Now, this is one down here. Imagine a donut that's been cross-sectioned and you're looking at the hole that goes down the middle of the donut. That's essentially what a channel protein looks like. They allow sugars and ions, so remember an ion is a charged atom, to pass through the cell membrane. Some are passive, meaning that they are essentially open for business. They let ions through whenever they want to come through, or sugar molecules to come through. They don't need any energy to work. Some channels are active, and that means that they do need energy to work. And if that's the case, we then call that channel protein a pump. Think of like a hand pump. If any of you have ever used a water pump where you actually had to move your arm and move the handle to get the water out. I've used one at a campsite before. That is kind of like what a channel that needs energy is like. The water is not going to come out unless you put some energy into allowing the water to come out. So then we call it a pump. This picture below shows exactly how channel proteins work. They have a hole. The molecule can move through the hole from either outside the cell in or vice versa. And then again, if you look really carefully, you can see the lipid bilayer. Channel protein there. And then solute, again, is the molecule moving through the membrane. Now, receptor proteins are the proteins that respond to chemical signals and trigger a reaction inside the cell. If you've ever heard of a hormone before, many hormones elicit a response by the cell by attaching to receptor proteins. Once the hormone is attached, it will cause a change inside the cell as a response. Receptors only fit certain molecules. So if, for example, that hormone attaches to the receptor, it binds and it triggers a response inside the cell. Examples of hormones that maybe you've heard before are adrenaline. Adrenaline can attach to a receptor, triggering a response inside the cell. Maybe you're nervous or you're scared. Your heart starts beating more rapidly, your respiratory system kicks in, and you start breathing more heavily. It's because adrenaline is triggering certain cells in the body to respond. Finally, glycoproteins, which are also called marker proteins, have a carbohydrate chain attached to the top of them, and those are shown here. So it's labeled kind of small. But the funny thing about this protein is there's actually a carbohydrate attached to the protein, which is how they get their name. Glyco means sugar, and then of course protein. Marker proteins or glycoproteins identify the cell that it belongs to you. If there's no marker or the wrong marker, the body could attack the cell with the immune system. It also helps with cell-to-cell interactions. Now, you might think you've never heard of this before, but I bet you have. If you've ever heard of blood type before, A, B, and O, these are markers. if I have a red blood cell and I am type A, it's because I have A glycoproteins on the surface of the red blood cell. If I have type B blood, the markers that I have are B. And if I have O blood, now things get interesting. O blood has no markers. And this is why, if you have O blood, you are called the universal donor. Oops. Sorry about that. You're called the universal donor because your blood cells can be given to different people and they do not recognize the blood as being foreign because there's no markers on it. But if you have B blood, you have to give B blood to other B people. If you have A blood, you have to give the blood to other A people. There's also another blood type called AB, and I'm not going to draw it, but those people have A and B markers. So they have to receive blood that is only AB. They cannot receive, or they can receive, sorry, let me draw that out. AB people can receive A blood, they can receive B blood, they can receive O blood, and they can receive AB blood. And so they are called the universal receiver. So that's what markers are. So just to recap, markers identify cells, blood type being a great example of marker or glycoproteins. Receptor proteins receive chemical signals and trigger a reaction inside the cell. Channel proteins allow molecules to enter and exit the cell, some being active and some being passive. And all of these proteins work in conjunction with fat and cholesterol to make up the composition of the cell membrane. So I'm going to scroll all the way back down to here and just kind of show you the picture because this shows you all of them together. And I'm going to highlight the parts you need to know. You need to know phospholipid, what the lipid bilayer is, cholesterol, which is kind of labeled over here. I'm just going to erase that. Glycoprotein, aka a marker, channel protein, and this we're just going to use the one that's always open as an example, and finally a receptor protein. You don't have to worry about gated channel or a transport protein right now. Looking at this picture, just know that this is a great example of a generic cell membrane. You can use this for any modeling activities that you get assigned in class. And that's it.